Hi students, here is the continuation of our study of the nervous system and senses. This narration will focus on the special senses, which are hearing, vision, taste, and smell. So this covers slides 28 to 34. Slide 34 is actually some questions. So we will begin with the air. So again, this course is called Anatomy and Physiology. So you are expected to be able to label a diagram of the air accurately. And as I go through these slides and this narration, I will try to highlight the important anatomical structures and the function that they perform. So we can divide the air into three parts, essentially. The outer air is what's referred to in this diagram as the external air. So we have an external part, the middle air, and the inner air. So which structures are in the external air? Well, here we have this part which we all recognize as our ear, the auricle, also called the pinna. This then leads to the passageway, which some of us like to push a Q-tip in our fingers, and that we're not supposed to do. So sound waves are collected. So sound travels in waves, which you might have learned in another class or maybe from physics. And so the sound waves move into the air and they will get to the eardrum, which properly is named the tympanic membrane. So it functions like a drum because when you beat a drum, you actually get a sound. So the sound waves vibrate. When they hit the eardrum vibrates because of the sound waves hitting it. And those vibrations are transmitted to the middle ear. So the middle ear contains the auditory ossicles. So know this word ossicles. They're the three tiniest bones in the body, actually. We do not list them when we list, some of the bones of the skeleton because we will discuss them here. Now, these three tiny bones in the body, they have names, and the sequence is important. So the first bone is called the malleus. The second is referred to as the incus, and they're all touching each other. And the third, which looks like stirrups, is actually called stapes. So those are the three bones, the auditory ossicles. And so the vibration from the tympanic membrane is transmitted first to the malleus, then to the incus, then to the stapes. Now the stapes is actually covering a structure called the oval window, not to be confused with the round window. So there's an oval window here, which is really an opening. And so the vibrations are now going to move into the inner ear. So what structures do we have into the in the inner ear? So far, all we've looked at is the movement or the passageway, the pathway of the sound waves. Before we leave the middle ear, I do want to point out this structure here, the eustachian tube, because this tube leaves, the middle ear, and it goes to actually an area at the back of the throat called the pharynx. So if you open your mouth, at the back of your mouth, that area there leads to an area called the pharynx. And the pharynx is actually open. Eventually you'll have to know this. The pharynx is a sort of fairly long passageway. And the upper part of the pharynx is actually open to the nasal passages. So back of the nasal passages lead into the upper part of the pharynx. You open your mouth, The back of the mouth leads to another part of the pharynx, referred to as the oropharynx. And then there's a lower portion which is closer to where the voice box is. So that area, the reason I'm mentioning this is because this area is shared. It is shared by the nasal passages, which are part of the respiratory system. It is shared by the oral cavity, which is part of the digestive system. And then we have the lower part, which is also part of the respiratory system. So what that means is bacteria, let's say if you have a throat infection, that the bacteria can actually migrate, move, climb, and go up to the nasal pharynx, pharyngeal region here. So the eustachian tube connects the air basically to the throat. Now in the inner ear we have the cochlea. So this is the structure that looks like a snail shell here, this coiled structure here. And the cochlea is the structure that has inside other structures that help with hearing. So there's fluid actually contained within the entire inner ear. So within the cochlea there's fluid. The vibrations will disturb that fluid, kind of the way, you know, waves on a beach sort of move. And that disturbing of the fluid causes an action potential to be set off, well, many action potentials, but they're all leading to this nerve here called the cochlear nerve. So the movement leads to an action potential, and then we have that information now being transmitted to the cochlear nerve. And the cochlear nerve will send that information to the lobe of the brain that's responsible for hearing, which is a temporal lobe. Other important structures in the inner ear are two structures that have nothing to do with hearing, but one of the other functions of the ear is equilibrium or balance. So you know, sometimes when you were a child, probably you would spin around, and when you stop, you feel dizzy. Well, the reason you feel dizzy is because the fluid that are now in this structure here. So there are 3 canals that are at 90 degrees to each other, and they have fluid in them. And so when you spin around, the fluid is also moving. When you stop, unfortunately, the fluid doesn't stop immediately. It still keeps moving. And so your brain gets this mixed message because your feet have stopped, that, oh, I've stopped. But then the semicircular canals here are sending the message because the fluid inside them is moving, right? They're sending the message that, oh, you're still moving. And that confusing sort of messaging is what leads to our sensation of dizziness. So the vestibule is actually a structure at the base of the semicircular canals, and information from these 2 structures are carried by a different nerve called the vestibular nerve. And some of that information will go, for example, to the cerebellum, because the cerebellum is responsible for helping us maintain balance and posture and equilibrium. So 2 very important functions to the ear. A little bit more on the vestibule. So here we see the cochlea, but that's not what we're talking about. These are the semicircular canals here. And so we have the vestibule is this enlarged. So at the base, there's a sort of an enlarged portion right there. So all of these names, you don't necessarily have to know. That's for a more advanced study. But endolymph is the fluid. So if you take a cross-section of one of these and you look at it, what you will see is there's fluid, and that's the fluid that moves. Now what we have in here is a structure that when the head tilts, so it tilts forward, gravity acting on it, or if the head starts moving in a straight line. So here is the head in a straight line, Here's the head tilting. What we have is these are some stones. They're actually made of calcium carbonate. They move, and when they move, the endolymph move. The endolymph is between there. And they're special cells. They're often referred to as hair cells. They are connected to the nerves. So these hair cells here, because the hair cells are being disturbed, they're no longer standing straight. They're now bent. That triggers an action potential. And that information, again, is sent to the brain, and the brain interprets it, or you know that your head is leaning forward, for example. The semicircular canals have more to do with rotational movement of the head. So moving your head essentially side to side, etc. And there's a different structure called the ampulla that's essentially responsible for that. And it's basically the same philosophy. You have endolymph. When the endolymph moves, it drags against, these are hair cells in here. And again, these hair cells trigger an action potential, which then is sent to the brain. So the minute details of all of this, we don't teach at this level. In a more advanced class, you'll learn that. And that takes us to the end of the ear. Now let's take a look at the eye. So again, we want to approach with the anatomy of the eye. You will need to know how to write the pathway for light entering the eye and the different structures that are important here. For vision, we will talk about again in sort of a general sense. Now, the type of receptors that we were just talking about when we look at the ear are referred to as mechanoreceptors. Mechano comes from the word like mechanical, and that's because those receptors, the hair cells, they respond to, the vibration, the movement of the endolymph. In the eye, we have photoreceptors. Photoreceptors, the word photo usually means light. So these are unique special receptors. They're only found in the eye and the human body, and they respond to light. And as you know, light has varying intensity. It can be dark, it can be medium bright, and it can be very bright. So let's take a look. The sphere of the eye can be divided into an anterior and a posterior chamber or compartment. The anterior cavity contains aqueous humor. Now, the word humor in biology means fluid. It's actually a very ancient word. But aqueous, I think if you've done enough science, you know aqueous usually means like a liquid, aqueous humor, while the posterior cavity contains vitreous humor. So let's identify the posterior cavity is easier to identify, and that is sort of this part here. This is folded in to show you, but that would be all of this kind of white area here. What most of us would consider kind of like the eyeball. All of this here is the posterior cavity, and vitreous humor is the substance that's in there. It's a more jelly-like substance compared to aqueous humor. Now the anterior cavity, which is shown at the front here, contains the aqueous humor, which is more liquid-like. Now let's go to the structures of the eye that are shown here. So these muscles, lateral rectus muscle, right? So this one and this one here, medial rectus muscle, I'm only going to point them out, but I'm not going to say anything more about them. There are 6 muscles that allow us to move our eyes up, down, left, right, diagonally, etc. And these are skeletal muscles. So you're shown 2 of the 6 on this diagram. More importantly, we want to start at the back of the eye and look at these layers. There are 3 layers of tissue covering the back of the eye. The first layer, the outermost layer, is called the sclera. The sclera continues to the front of the eye. And so when you look at someone and you see the white of their eye, you're actually looking at the sclera. It's a very tough, fibrous, strong kind of tissue. The choroid is the second layer. And the choroid has a lot of blood vessels. It's the nutritional kind of layer. And the third layer is the retina. The retina is where we find the photoreceptors. And the photoreceptors are called rod cells and cone cells. So I'm just going to clear the green that I put in here because we don't want to, there we go. So light coming in the eye, and I'm just going to draw a straight line, right? So light coming in the eye basically has to pass all the way through and come to the back of the eye here, because on the retina is where the image will be formed. We'll take a closer look at the retina in a minute, but we also have a nerve. So one of the things you should notice is that the special senses have very special nerves associated with them that are responsible for taking the information from these special organs to the brain. So the ear had the vestibular nerve and the cochlear nerve. The eye has the optic nerve. If you're wondering what the Roman numeral 2 is doing there, all of these nerves, if you see a Roman numeral within the name, it's because they're referred to as cranial nerves. And all the cranial nerves, there are 12 pairs of cranial nerves, all of them are usually numbered with a Roman numeral. So this would be cranial nerve number 2, or the other name is the optic nerve. So the optic nerve, once that image is formed at the back of the eye, the optic nerve sends that information to the occipital lobe. Because if you remember, the occipital lobe interprets visual information. So those are the 3 important layers at the back of the eye. Now, let's move to the front and see what happens. So the sclera continues to the front, like I said. If you look at someone and you see the white of their eye, you're looking at the sclera. But there's a transparent dome at the front of the eye, this very first part that light has to pass through, and that's referred to as the cornea. So I'm going to remove again some of my markings just to keep our image reasonably clear. I'll leave the cornea there because we have to come back to talk to that. There we go. Okay, so now we are looking at the cornea at the front of the eye. So the cornea is transparent so that light can pass through. It is true with age or cataracts that the cornea can actually become cloudy. The good thing is that the cornea in all humans actually does not have its own blood supply, which means that white blood cells, which are part of the immune system, don't actually migrate to the cornea at all. And so this makes it very easy to do corneal transplants, because I think you know that if you have to transplant tissue from one person to another person, normally you have to find a match. These people have to be genetically matched. With corneal transplants, they're one of the more easy, successful transplants to do. So that's the cornea. Now, So the cornea would be the first structure you would list in terms of listing the path of light through the eye. The next one you want to list is the aqueous humor, this chamber here that has aqueous humor. And that aqueous humor actually is refreshed many times throughout the day, because there's a little canal here which you can't really see well, but there's a little canal called the Canal of Schlemm, and that allows it to be refreshed. All of these fluids that we're talking about, whether it's synovial fluid in the knee or cerebrospinal fluid, in the brain. Now we're talking about aqueous humor. They are all derived from the liquid portion of blood. As blood circulates, these fluids, there's a mechanism to sort of generate these fluids and to refresh them. All right. So that's aqueous humor. So that would be number 2 on your pathway of light. Number 3 would be the pupil. And the pupil is an opening. So you can actually do a little experiment with somebody at home. If you're sitting in a room and you look at the person's pupil, switch off the light in the room. Let the room become very dark, and you will see the pupil dilating. The diameter of the pupil expands. And that's to allow more light in, because now you're in darkness, and the goal, of course, is to try and see as much as you can. It does take a while, and your eye will adjust, and eventually you will start to see. If you put the lights back on, the pupil will now reduce in diameter because now it's very bright. So that's the function of the pupil. It allows light in, but it also can change its diameter to regulate how much light is coming in. That ability of the pupil to change its diameter is due to the next structure called the iris. And so when you're looking at the pupil, you will see around the pupil of the person's eye, you will see what we often refer to as the color of their eye. But this structure that we point to and talk about as the color of the eye, that's the iris. The more important feature of the iris is that it is made up of smooth muscle. And these muscles are what control the diameter of the pupil. So the iris is really more important than just the, color of the eye. It's made up of smooth muscle, and it regulates the diameter of the pupil. So we don't put the iris in the path of light through the eye, because light isn't passing through the iris, it's passing through the pupil. Once it passes through the pupil, it gets to a really important structure, and that's the lens of the eye. So that's shown here. The lens of the eye has a number of interesting features, but before I say that, I want you to notice that it's attached to structures called suspensory ligaments on both sides. So the suspensory ligaments, as the name suggests, and also the ciliary body, ciliary muscle, these help to change the shape of the lens. So when we are looking at objects very close, the lens is very rounded. When we're looking at objects far away, the lens gets very elongated, like that. So these muscles in the ciliary body help to change the shape of the lens. This you should know and remember. That feature, the ability of the lens to change shape, is called accommodation. That's a word you should know. I'm sure it's in the lab somewhere, and you have some experiments to do in the lab, which you ought to do. It'll help you understand your eye better. Now, while there's this sort of, structure looking like feathers shown here, and this shows up in many diagrams, as you can see, there's no label to it because this isn't a real structure in the eye. This is very diagrammatic. I think it's probably just meant to show what a picture of the eye would look like if you had it in sagittal section like this. But immediately from the lens, the light will pass directly to the vitreous humor and then to the retina. So having the choroid, which is that layer there, having the choroid is like the back of this layer here. Having the choroid which is a pigmented layer, prevents light scattering inside the eye, because the image has to be formed on the retina. The retina is where the photoreceptors are. So let's take a look at what's written here. As the eye ages, these humors can dry and cause you to see floaters or specks of dust falling. When light is directed at the fovea centralis at the back of the retina. Okay, that's a really important location. So here's the fovea centralis. Let's follow it. And what this is saying is, at the back of the eye, here, there's a very special area where we find a high density of cones. Cones allow us to see color, distinguish color, whereas rods help us with black and white vision. Cones also help us to see details. And these two, the rods and cones, are the photoreceptors. They're not equally scattered around on the retina. The fovea centralis is a location where we have a high density of cones. And so when we're looking at an object and we can't see it clearly and we move our head, what we're trying to do is get the image to fall on the fovea centralis, right there. That will give us the clearer, more detailed image. Rods tend to be more in the periphery of the retina. Rods help us, like I said, with black and white vision. So rods help us with night vision too when light is low. And I think everything else that is sort of written here, I have told you. But let's take a look again. We see a central retinal artery and vein. It shouldn't surprise you. These are all blood vessels that are shown here. You know, these, what may look like lines, these are blood vessels in the eye. And yes, if you have type 2 diabetes, one of the problems you could encounter later in life, if your blood sugar is not well regulated, is blindness. So high amounts of glucose in the blood can damage blood vessels, and if you damage the blood vessels in the eye, then it will affect your vision. So there are many different diseases associated with the eye. The retina, for example, can become detached. This may happen in older people. It can also happen if you play sports and you get an injury, or if you're in an accident, for example. Then the lens, as we get older, the lens here also can get cloudy, but one of the more common problems is that these ligaments and the muscle for changing the shape of the lens during accommodation, they don't work very well, because as you get older, everything starts to work less well. And so most people find by about 40 years of age, they have to wear glasses. And that's called presbyopia. And that condition is due to the inability of the lens to change shape for, you know, close vision versus distant vision. So one of the, depending on the person and their eyes, you know, you might see them wearing reading glasses, for example. The optic disc, this is important. So the optic disc is commonly referred to as the blind spot, but please know the correct name. And the optic disc is this area here where the retina sort of does not continue. So the retina does not continue across this part here. Why? Because that's where the optic nerve exits. So there is no retina there. This is all now, nervous tissue. And so if an image falls here, I use a different color. If an image falls here, you won't see it. We cannot see it because there are no photoreceptors there. And you might be familiar with this because you're supposed to do a shoulder check, when you're changing lanes. And I think we've all done it sometimes. We start moving, then we signal, and then we check, only to find that there's a car, pretty close to us. So that's the optic disc. I think we have covered all of the structures, the important structures. So again, you are expected to be able to label this diagram and to be able to assign structures to the different functions. So one other slide, nerve impulses from the retina go to the optic nerve where visual information is sent through the optic tract. So that's what these the optic tracts, these blue and the red, is showing you. But what it's actually also showing you is the information from the left eye actually goes to the right side of the brain, and information from the right eye goes to the left side of the brain. But it's all going to the occipital lobe. Now for the last two, taste and smell. These are a little bit simpler. I think we all know that we use our nose, nasal passages. So high up in the nasal passage, so here we see the nose, nasal conchae are these shelf-like structures which have nothing to do with our sense of smell. But when we inhale air, it is kind of filtered through this structure. We might come back to that when we do the respiratory system, because air needs to be clean before it goes down to the lungs. So we have what is called olfactory epithelium. So if you ever notice that in order to smell something really well, you have to take a deep inhale. And the reason for that is you've got to send the odor molecules high up to touch basically this olfactory epithelium. So the olfactory epithelium is connected to an area called the olfactory bulb, but more importantly, we have an olfactory nerve, which is actually not shown in this diagram. Instead, we see the olfactory tract. Well, the olfactory tract will lead to the olfactory nerve. That is also a cranial nerve. So what happens is the odor molecules have to bind to the receptors in the olfactory epithelium. So let's say this is kind of, what a receptor looks like. Your odor molecules have to fit into this space here. So there's a physical binding of the odor molecules to the receptor. The binding triggers a nerve impulse, which all of the nerve impulses are collected by the olfactory nerve that is sent to the brain, and the brain interprets that you're smelling cinnamon instead of lemon, for example. One of the things about the sense of smell and taste is that these receptors are called chemoreceptors. C-H-E-M-O, with the word receptor written after it. Chemoreceptors. Why? Because the substance that you're trying to smell has to be dissolved in the mucus. Mucus is that slimy stuff that you might think is kind of icky, but it actually has a purpose. It helps to keep the epithelium of the nasal passages moist, and the odor molecules have to be dissolved in the mucus in order for us to smell. So both taste and smell use chemoreceptors. So if we take a look at the sense of taste, then we know that the tongue is covered with these small bumps which most often we refer to as our, taste buds. They're actually called papillae, and the papillae contain the taste buds. So the taste buds contain, again, special cells, gustatory cells. So gustation refers to always food, something to do with food. Gustatory cells that respond to chemical stimuli dissolved in saliva. So again, this is how chemoreceptors would work. So the different foods get dissolved in the saliva, and the gustatory cells, again, they are connected. Now, in this case, there isn't one nerve. There are actually several that are related. So that's why we don't give you the long names of the nerves. We just say nerves will bring the information from these chemoreceptors to the olfactory bulbs within the nasal cavity. And so we actually enjoy our warm food as opposed to cold food, usually because we can smell, as the vapor from the food is coming up to our nose, we can smell it. And it's not surprising that if you have a cold and a blocked up nose, nothing really tastes good. That's because you can't smell it. So these two are connected in terms of the interpretation, that our brain will give us, the feedback that our brain will give us. Now, different types of papillae are found in different regions of the tongue to detect salty, sweet, sour, bitter, and umami. So these are the 5 flavors. Now, you might have seen a tongue map, when you're Googling or searching or in past learning. The way that is presented most often is not correct. Because what the tongue map does is try to say, okay, this is the only region of the tongue where sweet can be detected. So the receptors, like for detecting sweet, is right here. And bitter is at the back here, and that's the only place. That's not true. So it is true that there might be a high concentration of certain receptors that detect a particular, taste, but most of the receptors are all over the tongue and not necessarily this high concentration in very specific areas. So this is the last slide, and I think I've given you enough information to think about, so have yourself a good learning experience.